Welcome to today's Wednesday seminar. In the spirit of reconciliation, let's all acknowledge the traditional custodians of the country throughout the reach and indeed well beyond the reach of this virtual seminar, and acknowledge their connections to land, sea and community. And we pay our respects to their elders, past and present, and extend that respect to all Aboriginal and Torres Strait Islander peoples today. So today's seminar is going to be presented by Marcel Doerflinger, who's a postdoc in my lab. And I find it always a bit of fun introducing great people because it gives me liberty to use tongue-in-cheek comments without, in this case, offending Marcel, who is a great scientist. So when Marcel spoke to me several years ago about wanting to do a postdoc in my lab, he said that he actually always really was keen to do a PhD and postdoc in Andreas Strasser's lab, and that I was actually the next best thing. Now, yeah, some people that would have hit him, but I actually took it as a compliment, and I think he made the right choice in choosing me because, one, I'm more stylish than Strasser. I certainly have fewer enemies, and generally I deal with super cool diseases, but I'll elaborate on that last point in a second. Now, despite my resolve to throw every conceivable project from basic science to translational work the way of Marcel, in his heart, he truly pined for Strasser. And even though there was a 1.5 metre thick block of concrete between level 4 and level 5, that didn't stop Marcel from falling in love with a project that Marco, Harold and Strasser were running. And the real irony is that it comes to the point that I made before, was that the project they had for him was actually to do with a super cool disease, in fact, an infectious disease. So I've never quite figured out if all this happened because Marcel was desperate to work with Strasser or because Strasser and Marco were desperate to work on a super cool disease. But what happened was a brilliant collaboration engineered by an insightful, truly meticulous and incredibly passionate postdoc who followed his dream of working with Strasser despite all the obstacles I created. So Marcel, you've done a phenomenal job on this project, so carefully crafted, and we all look forward to your presentation that is a true testament to your collaborativeness in spanning divisions of WEHI, but actually also across the road to Micro-Uni Melbourne, where you've collaborated with Sami Badawa. Now hand over the Ethernet to Marcel.
Yeah, thanks, Mark, for those warm words. I'm not sure if I really like Andreas more than you, but we can discuss that afterwards. Welcome everybody to the seminar today. I hope you're all safe and sound during this challenging time. And of course, it was a dream to come to WEHI and work on cell death, and it was also a dream to give a Wednesday seminar, despite maybe not in these circumstances. But anyway, it's my great pleasure and my honor to talk to you today about the fascinating and very flexible ways our immune cells have found to kill themselves in order to protect us from intracellular pathogens. Now, almost 200 years ago, Carl Vogt was the first to report his observations on dying cells in toads, of all things. And it was Walter Fleming who, looking down his microscope, noticed interesting spontaneous cell death that was morphologically different from necrosis in the late 1880s. But it took almost a century until Lokshin and Kerr first described what they thought would be a programmed cell death. And then, shortly thereafter, two giant leaps for mankind occurred. So humans flew to the moon, and the term apoptosis was coined. And I personally find it very fascinating that humans were flying in space before we even knew about this fundamental process that shapes so many cells in our bodies on a daily basis. So over the course of the next decades, of course, WEHI was contributing big strides towards our understanding of apoptosis, which was for a long time considered to be the only form of programmed cell death. Until the early 2000s, when pyroptosis was described, a morphologically quite different type of cell death. And in 2005, necroptosis joined the family. And then in the following 15 years, a variety of new forms of cell death, including ferroptosis, was described. And it almost feels like a new form of cell death was popping up like mushrooms every other year. I did a PubMed search just the other day, and the exponential growth in published work really highlights the importance of this research field. And it almost looks like the coronavirus curves we are so used to looking at these days. Now, we could ask the question, why are there so many ways to die? And I think it's quite evident that cell death has to be very tightly regulated because it has such a central role in maintaining healthy physiology, for example, in regulating development and preventing autoimmunity, but also in disease progression, such as in diseases such as cancer, heart disease, or neurodegeneration. But for most of my talk, and thereby, of course, pleasing Mark, I'll be focused on the important role cell death has in immunity and infection. So programmed cell death is an important means in our fight against pathogens, as for example, cytotoxic T cells can induce the killing of infected target cells. But cell death also in general is very important in shaping our immune cell repertoire, for example, by negative selection, and the contraction of immune cell populations by apoptotic removal of pathogen-specific T cells, B cells, or antigen-presenting cells is critical in terminating our immune responses. However, too much immune cell death can also be detrimental, as for example, HIV-induced killing of CD4 T cells or sepsis-mediated lymphopenia can lead to immunosuppression and leaves the patient vulnerable to secondary infection. And lastly, cell death of infected phagocytes has an important role in bridging the innate and adaptive immune responses by activating antigen presentation or by removing the pathogen's replicative niche and re-exposing it to other components of our immune system. So really cell death goes full circle when it comes to fighting the pathogens. So cell death can broadly be classified and divided into two main types based on their morphology, into lytic cell death, such as pyroptosis or necroptosis, as well as non-lytic cell death, such as apoptosis. And these two cell death forms are very different in their characteristics. And while lytic cell death leads to spilling of intact cellular contents, apoptotic cell death actually has very different immunological consequences because the cellular contents are degraded in a systematic way. And while lytic cell death is highly inflammatory, leading to the recruitment of more immune cells to the site of infection, apoptotic cell death, on the other hand, is considered to be immunologically silent and enables uptake and presentation of antigen. So in general, lytic cell death can therefore be considered less beneficial to the host than non-lytic cell death. And although you can appreciate that these two types of cell death are morphologically and in their consequences very distinct, they're both initiated and regulated by proteins called caspases. Now, caspases can therefore be classified either based on the type of cell death that they're known to be engaged in, for example, pyroptosis and apoptosis, but also based on their structure and function. They can be classified into initiator or executioner caspases of cell death. So activation of the initiator caspases occurs via so-called multi-protein adapter complexes, such as the inflammasome, the disc complex, or the apoptosome. And binding with the initiator caspases, caspase 1, 8, or 9, leads to their homodimerization and autocatalytic cleavage, which release active fragments that will go on to induce downstream cleavage of their respective effector molecules, leading to pyroptotic or apoptotic cell death. Okay, so now we've covered the basis. Let's have a closer look at the individual cell death pathways. So inflammasome activation by either danger or pathogen associated molecular patterns, or LPS, will lead to dimerization and cleavage of the inflammatory caspases 1 or 11, which leads to downstream activation and cleavage of gasdermin D, which is the pyroptotic effector protein, which goes on to pour forms in the outer membrane. This leads to rupture of the cell and release of cytokines processed by caspase 1, such as IL-1 beta, and to an inflammatory cell death. Apoptotic cell death, on the other hand, can either be induced via the intrinsic pathway, where dysregulation of members of the BCL2 family member of proteins leads to permeabilization of the outer mitochondrial membrane, apoptosome-mediated cleavage of the initiator caspase 9, and subsequent cleavage and activation of effector caspases, leading to catastrophic proteolysis and a systematic degradation of the cellular contents, as I described before. Now, alternatively, death receptor ligation can lead to cleavage-induced activation of caspase 8, which in turn directly induces the effector caspases, or can cause mitochondrial damage via BIT, followed by a caspase cascade induced by caspase 9. So attempts in the late 90s to delete caspase 8 in mice revealed embryonic lethality upon caspase 8 deficiency. And it wasn't until more than a decade later when it was revealed that caspase 8 actually negatively regulates necroptotic cell death via inhibition of RIPK3, which in itself is the upstream regulator of the pore-forming protein MLKL, which induces necroptosis. So you can see, therefore, non-lytic cell death via death receptor-mediated apoptosis and lytic cell death in the form of necroptosis were already shown to be intimately linked via caspase 8. However, for many, many years, apoptosis and pyroptosis were thought to be individual entities with independent activation mechanisms and distinct physiological outcomes, as previously described. And while the long-standing dogma in the field was that, well, the inflammasome initiates caspase 1 and death receptor signaling activates caspase 8, recent evidence based on an increasing body of literature using in vitro studies indicates that indeed the inflammasome can also lead to activation of caspase 8, resulting in both apoptotic downstream signaling, as we would expect, but also in proteolytic cleavage of Cas-domin D and ensuing pyroptosis. So really only in the last few years, and while our studies were already underway, a certain degree of overlap of individual components of not just necroptosis and apoptosis, but also of pyroptosis and apoptosis has become increasingly appreciated. Now, as pyroptosis, necroptosis, and apoptosis have all been shown to be implicated in host defense against infection in one way or the other, this led us to the question that we wanted to see what the relative contribution of individual caspase-mediated cell death pathways was to host defense against intracellular infection in vivo. And to this end, we started a collaboration with Sami Bedoui from the Doherty Institute across the road. And he's an expert in using salmonella as a model for intracellular infection. And salmonella is a key global cause for diarrhea, but it can also develop into typhoid fever, a life-threatening systemic infection that results in more than 100,000 deaths per year. And although there's a vaccine available, this does not confer 100% protection. And moreover, 5% of infected people become chronic human carriers. And due to the ineffectiveness of current antibiotic treatment and an ever-increasing antimicrobial resistance, this pathogen remains a global health problem. But salmonella also has its favorite host cell, which are phagocytes, and uses them as replicative niche to invade the host's immune response. So we thought we had an ideal tool to study the role of cell death in host defense against infection. So in the in vivo model, Sami's group has established and is utilizing since a few years, spleen and liver are major replication sites in mice that have been intravenously infected with a mouse-adapted strain of salmonella. And the bacterial burden reaches a peak in wild-type mice at around week 3 post-infection. And wild-type animals are then able to clear the pathogen over the course of the following weeks, as you can see here. So this animal model therefore enables us to decipher both the contribution of the innate immune response to bacterial control, as well as the adaptive immune response to bacterial clearance. So now, using gene-targeted animals harboring deficiencies in individual or combined cell death pathways, we therefore had an elegant in vivo model to interrogate the relative contribution, as I said earlier, of all major forms of cell death on bacterial control when looking at bacterial burdens in both liver and spleen. So when we infected mice that were deleted for the inflammatory caspase 1 and 11, and had a look at bacterial burdens in spleen and liver at 3 weeks post-infection, we could only identify a quite minor impact on the ability of the mice to control this bacteria. And the animals, as you can see here, were also able to clear the infection over the course of the following weeks, almost identical to their wild-type counterparts, indicating that other forms of cell death are able to contribute to bacterial control in vivo. Now, additional deletion of the quite dubious inflammatory caspase 12, which had previously been linked and implicated to ER stress-mediated cell death, revealed no additional impact on bacterial control to loss of the inflammatory caspases 1 and 11. And this was in line with previous study by the former PhD student in Marco's lab, Rania, who could show no contribution of caspase 12 to cell death and sepsis. So similarly, when we additionally deleted the necroptotic arm of the cell death response by deleting RIPK3, we could again only detect yet minor increases in the bacterial burden in spleen and liver over and above the other knockouts or wild-type animals. In mice that were deficient for caspase 8 and RIP3, and therefore deficient in the extrinsic and intrinsic extrinsic pathway of apoptosis and necroptosis, again, only a slight increase in the bacterial burdens could be detected. And please note that, as I previously mentioned, caspase 8 deletion on its own is embryonic lethal, and we therefore had to delete the necroptotic arm to study caspase 8 deficiency. However, finally, combined deletion of all inflammatory caspases, as well as caspase 8 and RIP3, led to a significantly increased number of bacteria in spleen and liver. That indicates that these animals had problem in controlling the infection over and above wild-type mice, or mice that lack only individual components of the cell death machinery. And most strikingly was when we left these animals on the shelf for a bit longer, the mice became sick around 4 to 5 weeks post-infection and had to be euthanized due to ethical reasons. And the bacterial burdens in both the spleen and the liver were significantly increased, which indicates that bacterial control broke down entirely when these three major forms of cell death were absent. So we speculated that this was due to an inability of phagocytes to undergo programmed cell death and to thereby purge the bacteria from the intracellular niche and re-expose them to the immune system. As I said, because macrophages are the favorite host cell for these bugs. And to this end, we used bone marrow chimeras, where hematopoietic stem cells from wild-type or gene-targeted donor mice are used to reconstitute the immune system of non-lethally irradiated target mice, recipient mice. And strikingly, what you can see here, animals that were reconstituted with the bone marrow from mice that were deficient for the inflammatory caspases 1, 11, 12, as well as caspase 8 and RIP3, had severe defects in controlling the salmonella pathogen, with bacterial numbers in spleen and liver almost similar to the full body knockout animals as I shown previously. So this really underlines the importance of cell death induction in immune cells as an important means to fight intracellular bacteria. So taken together from these in vivo studies, I could show you that cell death seems to play a central role in the fight against intracellular pathogens, and that combined deletion of all major forms of cell death pathways leads to failure of bacterial control in vivo. And our data really strongly supported previous in vitro reports that there's some degree of functional redundancy. So we wanted to see how this functional redundancy really looks at the cellular molecular level and to which degree. And we therefore turned to in vitro models. So bone marrow-derived macrophages are a widely used tool to unravel the molecular components of cell death during intracellular infection of phagocytes. So I'll talk you through this piece of data slowly, as you will see this kind of graph reoccurring throughout the course of my talk. So this graph shows the percentage of live cells over time after infection with salmonella as measured by the release of LDH, which is a surrogate marker for metabolic activity and thus the amount of live cells present in the well. So when we infected wild-type BMDMs with salmonella, a rapid cell death response is induced, and after 2 hours, already more than half of the cells have died, as seen by the steep drop in the survival curve. And the type of cell death appeared to be quite lytic, as shown by bright field microscopy. But as Kelly Rogers mentioned in her presentation last week, WEI has a world-class imaging facility. So I teamed up with Niall to look at the dying cells using our novel lattice light sheet microscopy. So I point you towards this picture here. So in green were GFP harboring salmonella, and this cell here is a really blown up, swollen cell in the middle, and that seems to display pores on its surface that are classical features of pyroptosis. And you can see that the nucleus has already taken up PI. So again, now we're zooming in. This cell is really swollen up, has all the hallmarks of lytic cell death, and you can see the pores forming. So there's a salmonella bug inside, and the nucleus has taken up PI already. So I really think you can appreciate that this seems to be a quite lytic cell death, and again, there's only a handful or a couple of bacteria inside. So morphologically, the rapid cell death appeared to be lytic, which indicated cells might activate caspase 1 via the inflammasome to induce gasdermin D-mediated pyroptosis, as I've shown you before. So that's a phenomena that's been widely described throughout the literature since discovery of this type of cell death. And sure enough, at the protein level, as shown here by Western blot, we see hallmarks of pyroptotic cell death, including the cleavage activation of caspase 1 and ensuing cleavage of gasdermin D. Now, when we additionally deleted caspase 8 and also RIPK3 due to the above-mentioned lethality, no change in the kinetics of the cell death that we saw was detected. So this indicates that inflammasome activation, albeit recently shown to be able to induce caspase 8 cleavage as well, can kill cells exclusively via caspase 1 mediated pyroptosis. However, BMDMs from mice with deficiency in the inflammatory caspases 1, 11 and 12, they died with a delayed kinetics, and we attempted to identify the exact molecular programs that were responsible for this cell death response. And as previously mentioned, recent studies suggested a certain degree of functional overlap with caspase 8, so we had a closer look at all three major forms of cell death as possible backup mechanisms for caspase 1,11 mediated pyroptosis. So firstly, necroptosis, which is another form of inflammatory and lytic cell death, was not responsible for the delayed killing, as we could not detect phosphorylated MLK, which is a hallmark for necroptotic signaling and key to the induction of the pore formation by MLK. So as a result, it seemed that pyroptosis is not backed up by another inflammatory cell death type, necroptosis. So that left us with the option of pyroptosis being backed up by caspase 8 mediated apoptosis or by caspase 8 mediated cleavage of gasdermin D. But we figured out the best way to look at that would again be using lattice light sheet microscopy. So together with Niall, we produced these beautiful pictures, and you can see here in the front there is a dying cell that is displaying all the hallmarks of apoptosis, such as membrane blebbing and the uptake of annexin and PI, indicating that there's already fragmented DNA in the blebs as well. And here in the background is a cell that is still at an earlier stage of apoptosis, where the blebs have started to form, but PI does not seem to have integrated. And keep in mind that the bugs, the salmonella bugs in this case, are shown in purple. So as you can see, again, this is a late apoptotic cell with nice apoptotic bodies formed. And in the back, this cell is at an early stage, and you can see that there's a remarkably higher amount of bacteria in those cells already as well. So here you can see the nucleus and the apoptotic blebs, and this cell is at an earlier stage of apoptotic cell death. So clearly, we think we could show that loss of caspase 1,11 is backed up by caspase 8 mediated apoptosis, and indeed this is the case. So there is no cleavage of gasdermin D detected, but strong activation of caspase 8 and all downstream effectors, including TBID and the effector caspases. So taken together, it appears that rather another form of lytic cell death, such as necroptosis or caspase 8 mediated apoptosis, acting as a fail-safe system for pyroptosis induced by the inflammatory caspases, caspase 8 mediated apoptosis kills the infected cells. And when we had a look at BMDMs from our quintuple knockout mice, lacking the caspases 1, 11, 12, as well as caspase 8 and RIP3, these cells were unable to kill themselves when infected with salmonella, even after 24 hours post-infection. And this really nicely correlated with our in vivo data, where I showed you just before that these mice completely failed to control infection. And again, lattice light sheet microscopy of these cells with the help of Niall revealed that these cells were completely unable to kill themselves. They were really filled up almost to the max with bacteria, as shown here again in green. So there's an actual live cell, and this one here as well, and this one as well. So they are kind of bubbled up, full with bacteria, but they cannot kill themselves because we cannot detect any PI or annexin-5 uptake. All right, so to sum up the in vitro studies, it appears that there is indeed a functional overlap in place between pyroptosis initiated by the inflammatory caspases, as well as caspase-driven apoptosis to ensure killing of infected cells. And this shows that the host can flexibly deploy different types of cell death for pathogen control. And we wanted to gain further insights into how exactly this functional overlap is regulated. Now, primary BMDMs are a bit of an effort to grow because they require a lot of mice, of course. And we therefore turn to immortalized BMDMs, which are easy to grow, divide fast, and also have been extensively used to characterize the host response to pathogens, including Salmonella. But more importantly, they can be genetically manipulated. And this is where Yeshua, a fantastic student in Marco's lab, got involved and initially helped me in generating these iBMDMs. But as you will see throughout the talk, this was an absolute mammoth task, and she took over the production line, so to speak, super efficiently. And we quickly developed a great collaboration where she generated cell line after cell line of knockout cells, and her contribution to this project, as you will see, was absolutely central to its success. And Yeshuan always said, we are a great team. I generate cells and you kill them. So thank you, Yeshuan. She's currently back in China, but she said she would try and hack the system there to watch this talk. So if you hear this, Yeshuan, greetings to China. And while wild-type iBMDMs undergo cell death with similar kinetics to their primary counterparts, additional deletion of caspase-8 and DRIP3 also had no effect on the cell death response in those iBMDMs as seen in primary BMDMs. Combined deletion of the inflammatory caspase led to a delayed cell death, exactly as seen in the primary cells. And finally, the combined deletion of all inflammatory caspases 1, 11, 12, as well as caspase-8 and RIP3, recapitulated the profound resistance to cell death as seen in primary cells derived from our knockout mice. However, one of course could argue that these cells, as the name rightly suggests, as they are immortalized, could be problematic when studying cell death of all things. However, the intrinsic apoptotic pathway should remain intact, and we therefore expose the cells with etoposide or BH3 mimetics, which are known quite exactly how they induce mitochondrial stress and induce downstream effector caspases to cause cell death via intrinsic apoptosis. And indeed, as indicated here on the right-hand side, immortalized cells deficient for caspases 1, 11, 12, 8, and RIP3 died in a similar fashion to wild-type cells, both upon treatment with BH3 mimetic cocktail, as well as etoposide, which really indicates that the downstream effectors of programmed cell death appear to be intact and that these cells can actually indeed die. So I hope I could convince you that these immortalized BMDMs are a great and versatile tool to study the interplay between initiator and effector caspases during cell death, during infection, as well as we could gain insights into the exact kinetics with which these molecules are being employed by the infected host cell. All right, so far we know that the inflammatory caspases, caspase 1, caspase 11, cause pyroptosis via gasdermin D, and we discovered a fail-safe system in which caspase 8 can replace the inflammatory caspase 1 and 11 by inducing apoptosis, or maybe even, as the literature suggests, can potentially induce gasdermin D to ensure cell death. So during one of our regular meetings up in the WeHi tea room, and I missed the tea room like many others of you, while Marco probably had his 7th cup of coffee and maybe while munching on a donut, just like Homer Simpson, because Marco likes donuts, we concluded that removing gasdermin D, as well as caspase 8 and RIP3, should prevent the cells from killing themselves upon infection. So we first infected gasdermin D deficient iBMDMs with Salmonella, and they died in the expected delayed manner. We assumed that as pyroptotic cell death was disabled and the macrophage instead induced alternative pathways towards cell death, presumably via caspase-8 mediated apoptosis. So, however, when we additionally deleted caspase-8 and RIP3, much to our surprise, the cells died in a way that was almost indistinguishable from the gasdermin D deficient cells that I just showed. And this indicated to us that there have to be yet other mechanisms of functional overlap in place, and that the inflammatory caspases 1 and 11 might be able to induce a non-pyroptotic cell death in the absence of gasdermin D. In order to define which of the inflammatory caspases 1 or 11 appears to be responsible for this non-pyroptotic cell death, independent of gasdermin D and independent of caspase 8, we additionally deleted caspase 1. So given that this completely abrogated the cell death response, as seen here, we concluded two things. So first, caspase 11 can only kill cells exclusively via gasdermin D, and this confirms some speculation in the literature as to what extent caspase 8 can back up for caspase 1. But more importantly, this showed that caspase 1 is the driver of this backup system, because cells absolutely fail to kill themselves when we delete caspase 1. And using immunoblot analysis of cells deficient for gasdermin D, caspase 8 and RIP3 confirmed this hypothesis, as we could see strong cleavage and activation of BIT over time, indicating that caspase 1 indeed can induce cell death via the mitochondrial amplification loop, leading to activation of downstream caspases such as caspase 9, 3, and 7, and PARP, which is a marker for DNA cleavage, as shown in the Western blot here. Now, interestingly, while we were making these discoveries in pathogen-infected cells, a Japanese group published similar findings using an artificial caspase 1 dimerization and activation in vitro system. So even though I first thought we just got scooped, it at least confirmed our findings that caspase 1 has the capacity to not just induce pyroptosis, but to also induce apoptosis via BIT-mediated induction of mitochondrial damage. So we convened in the tea room again, discussing these findings, and decided to prevent cells from inducing caspase 1 mediated apoptotic cell death. We have to block the mitochondrial amplification loop by deleting BAX and BAC to provide further genetic evidence to our hypothesis. So, however, as you can see in the graph quite clearly, additional deletion of the inducers of mitochondrial pores, BAX and BAC, did not prevent cell death from occurring in salmonella-infected cells. So cells deficient for gasdermin D, caspase 8, RIP3, as well as BAX and BAC, still died with a similar kinetic. And this strongly indicated that cell death via caspase 1 can happen independently of gasdermin D, or even independently of mitochondrial outer membrane permeabilization. And we hypothesized that this may occur via direct activation of the executioner caspases 3 and 7, or by activation of caspase 9. And this was the case, as immune blotting revealed induction of all downstream caspases as well as PARP cleavage, independent of the ability of BIT to cause mitochondrial damage via BAX or BAC. And I want to point your attention to caspase 9, as activation of caspase 9 appeared to be slightly delayed in these cells compared to cells that still harbor BAX and BAC. This indicated to us that indeed caspase 1 may activate caspase 9 directly, which can then activate the downstream effectors to induce cell killing. So you might have guessed it from knowing how regular Marco was to be found in his bill of Tiro. It was time to reconvene again and discuss these novel findings. And of course, we decided to keep striking while the CRISPR iron was hot, and additionally deleted the executioner caspases 3, 6, and 7 to finally write up our manuscript, because we increasingly got the impression that this was becoming a really hot topic in the field. But intriguingly, additional deletion of all downstream effectors still did not render those cells profoundly resistant to salmonella-induced cell death. It only slightly delayed the kinetics, but those cells were still able to kill themselves. And as we still saw a strong induction of caspase 9 in our infected cells, this, however, opened the exciting possibility that maybe caspase 9 can act as an executioner caspase, rather than just being an initiator caspase. So maybe caspase 9 can kill the cells. And finally, indeed, additional deletion of caspase 9 on top of all the other known executioner proteins reproduced the cell death response that we saw when all inflammatory caspases 1, 11, 12, as well as caspase 8 and RIB3 were missing. So this confirmed the unexpected finding that caspase 1 can indeed act as an executioner caspase to kill cells when activated via caspase 1. Okay, so to sum up this part of the talk, we had identified that caspase 1 can induce a range of processes to induce cell death with remarkable plasticity. It can induce pyroptosis via the cleavage of gasdermin D. It can induce apoptosis by bypassing caspase 8 through the mitochondrial amplification loop. But it can even circumvent the need for permeabilization of the mitochondria by directly activating the caspases, effector caspases 3, 6, or 7, or by activating caspase 9 directly. And a quite unexpected role for caspase 9 as an executioner caspase via caspase 1. And so while we were discussing our manuscript and being really happy of how we discovered the weird and wonderful ways the initiator caspases can back up each other and how caspase 1 can kill infected cells by apoptosis and not just by pyroptosis, we concluded that the deletion of all executioner of cell death should recapitulate the same phenotype seen in cells that lack caspases 1, 11, 12, as well as caspase 8 and RIB3. So we went back to the lab, but to our surprise, or maybe not to our surprise, because we were getting used to seeing unexpected cell death phenotypes, cells deficient for all known downstream effectors of pyroptosis, apoptosis, and necroptosis still died, although with delayed kinetics. And this indicated that there might be even more yet to be discovered backup mechanisms in place. So the question was, how did these cells still kill themselves? I mean, we saw strong activation and cleavage of caspase 1, and to a similar but minor extent, activation and cleavage of caspase 8. And so review of the literature suggested that possibly gasdermin E, which is a new kid on the gasdermin block, could be activated by caspases to induce pyroptosis, or that maybe ER stress plays a role, because it was shown to have an effect on host pathogen interactions during bacterial infection. So I explored these possibilities, but as seen on this Western blot here, neither gasdermin E cleavage nor the induction of ER stress, as shown by the ER stress marker BIP, could be detected. So looking under the microscope, morphologically the dying cells lacking all effector proteins or all known effector proteins appear to be apoptotic, which was puzzling because we were really sure that we deleted all the known apoptotic effector molecules. So I could finally convince Marco to do one of my favorite things, to get to the bottom of the questions if there's even more backup and what these executioners are. It was to perform a CRISPR screen. So to this end, I transduced IBMDMs deficient for all known executioners of cell death with a guide RNA library, and thereby generated a pool of cells in which individual clones were numbered by individual covering the whole genome. These cells were infected as previously with salmonella or left untreated as a control. So I repeated the infection process 3 times to induce a really stringent killing of cells and enriched the surviving clones and subjected them to next generation sequencing with the help of Stephen Wilcox in the genomics lab. So with this screen, I was hopeful to identify upstream regulators of infection and cell death, but most importantly to uncover novel backup cell death executions that drive salmonella-induced cell death in those cells. All right, so on the left-hand side, here you can see the top 50 enriched guide RNAs that mediated survival in salmonella-infected cells compared to the non-infected full library. So amongst the top hits was a range of host genes that had been previously reported to be utilized by the salmonella bacteria to gain entry and successfully infect host cells. And this nicely confirmed that our screen setup was able to detect critical host genes. So for example, host lipid rafts have been reported to be central to the ability to the bacteria to dock onto the surface and to internalize into their replicative vacuoles into the host cells. Similarly, the so-called wave complex has been shown to be exploited by Salmonella in order to manipulate the cytoskeleton and to enhance invasion. So given that our screen was able to reveal upstream host factors and even some novel effectors and interesting pathways I don't have time to talk about today, we were hoping we could answer the question of how these cells were still able to die. And indeed, most strikingly, our screen revealed as the top hit guide RNAs by far the inflammasome component NLRC4 as well as caspase-1. So this clearly indicates that caspase-1 activation via the inflammasome was absolutely critical and central to driving the cell death we saw in the cells that lack all the downstream effectors. So, and despite the fact that we couldn't really answer with this what the potential effector molecules might be, at least not till this point, this would also mean that when we block caspase-1 activity, the cells should not be able to kill themselves anymore and resemble cells that lack all initiator caspases. So to this end, I used VX765, a potent and very specific inhibitor of caspase-1 during the infection process. And as you can see here, the inhibitor indeed completely blocked the cell death response. This underlines the critical role for caspase-1 in inducing cell death of infected cells, even in the absence of all executioner proteins. And when I had a closer look at what was going on at the protein level, treatment of cells with VX765 indeed blocked caspase-1 activity, but surprisingly also strongly dampened the induction of caspase-8. So we therefore concluded that caspase-1 acts upstream of caspase-8 to induce cell death in the absence of all known downstream effectors, and that the inflammasome activity alone is not sufficient to fully activate caspase-8 in infected cells. That really caspase-1 is the main driver of this death response. And indeed, similar to inhibition by inhibitor treatment, VX765, in cells in which we additionally deleted caspase-1 could not undergo cell death anymore. And finally, genetic deletion of caspase-8 had identical effects than deletion of caspase-1. So this clarifies that caspase-1 indeed acts upstream of caspase-8, but also requires caspase-8 activity to induce cell death in the absence of all known downstream effectors. So caspase-1 and caspase-8 are therefore the critical upstream initiators, caspases, that drive cell death of salmonella-infected cells, but they can also act as executioner caspases in this case. Okay, so I hope that I could convince you that the ability to undergo cell death is critical to ensure bacterial control during salmonella infection in vivo, and that there's an extensive backup or fail-safe system among the initiator caspases to ensure that cell death is happening. Furthermore, our work has uncovered exciting new findings as to how caspase-1 can induce apoptotic cell death independent of gasdermin D, independent of caspase-8, independent of the mitochondria, and independent of the executioner caspases 3, 6, and 7. And that even more surprisingly, caspase-9 might act as an executioner when activated by caspase-1. And finally, we can show that cell death is even possible in the absence of all known downstream effectors, as long as caspase-1 and caspase-8 are present. And this, of course, poses the question we haven't been able to answer yet, but are now most interested in. How can caspase-8, with the help of caspase-1, kill infected cells? And what are its downstream effectors? So we are planning on tackling these questions in the near future, using proteomic approaches to identify novel caspase-8 targets, for example. But to come back to more like the initial question that I posed at the beginning, why is there so many ways to die? And I'll put on the theorist hat in the spirit of Phil Hodgkin a few weeks back. You know, maybe once the inflammasome is activated in infected cells, it drives cell death via caspase-1 and caspase-8 by any means. And given the extensive functional overlap which we have shown between initiators and effectors of several death pathways, rather than individual cell death pathways being in place, we are talking about one big overlapping network of cellular suicide that is being induced. And along this line, it's also tempting to speculate that cell death likely occurs simply based on which effector proteins can kill the cell first, rather than the ensuing cell death being a result of which effectors are switched on. So the whole system is switched on, and it's really a matter of who kills the cell first. And furthermore, our data may suggest that caspase 1 and caspase 8, and seemingly maybe even caspase 9, are both initiators and executioner caspases. And I'm tempted to use and calling the term master caspases, as they can flexibly deploy components of the cell death machinery, almost like puppet masters. And going back to the apoptotic function of caspase 1 that we have discovered, and bearing in mind that the very different immunological outcomes of lytic versus non-lytic cell death, it might be that maybe apoptosis rather than pyroptosis is the preferred outcome for the host upon inflammasome activation in vivo. Or maybe that both pathways are flexibly induced by caspase 1 and caspase 8 for that matter, to ensure that the pathogens are both purged from their replicative niche via pyroptosis, and to recruit more immune cells if needed, but also dismantled for antigen presentation. So really depending on the tempospatial status of the infection, so to speak, to fight the infection by any means. And maybe we have so far just been lacking the genetic models to dissect and understand this. So late last year, a critical role for caspase 8 as the molecular switch between the main cell death pathways has been proposed based on the central role it has in embryonic development. So we think that our work, as well as work by other groups published in a recent wave of very similar papers, suggests a similar role for caspase 1 as master caspase that orchestrates the cell death response in control to intracellular infection with great plasticity. And kind of trying to answer the question of why so many ways to die again, this time with the theorist head of Charles Darwin, is that maybe we have evolved so many ways to die simply based on the theory of coevolution with pathogens for thousands of years in a molecular arms race to determine the survival of the fittest. And indeed, this is reflected by the fact that both the host has so many ways to kill itself and seemingly flexibly can use them. Pathogens such as this bespoke Salmonella have evolved effector proteins that aim to counteract the attempts of the host cell to induce killing at many different crossroads of the cell death machinery. So it thus seems plausible that while the Salmonella intruder has found ways to manipulate our cell death machinery, we have evolved caspase superheroes that defend us against those bad boys, and that caspase 1 appears to be the last man standing. So where do we go from here? Of course, phagocytes are the host cell reservoir for many other intracellular pathogens, such as parasites like Toxoplasma or Leishmania, fungal infections including Cryptococcus and Candida, and a range of other bacteria. So all these pathogens have evolved to enable them to evade the host cell death response in very specific ways that are maybe different to what Salmonella has evolved. And we have generated throughout this project a big library of macrophages that lack cell death initiators and executioners in a broad variety of different combinations. So this cell death library will be very helpful in deciphering host pathogen interactions underlying these different infections further. And of course, we're happy to share this resource to anyone at WEHI or beyond if you're interested. Feel free to get in touch. But similarly to looking at different cell death processes in the Petri dish, we can make use of the now vast collection of mice deficient in individual caspases or combined cell death pathways in mouse infection models that have been established across the Parkville campus. And just to give you a little teaser, I've recently started collaborations across the infection division here at WEHI using different in vivo infection models with pathogenic parasites relevant to humans. So using our knockout mice, this data shows that mice infected with Leishmania, as well as Toxoplasma gondii, and also Chris Tonkin's new favorite parasite, Cryptosporidium. That all mice that lack the inflammatory caspases, as well as caspase 8 and RIB3, they all display difficulties in pathogen control when infected. So this certainly opens up interesting projects in which we want to shed light on which individual cell death pathway is important to fight these parasitic pathogens. So as you could imagine, it took several years to establish the mouse colonies harboring knockouts in combined cell death pathways that we have used for this project. And you might remember the data I showed early in my talk on how we could utilize bone marrow chimeras to show that cell death in immune cells, or more specifically phagocytes, is responsible for controlling salmonella. So the Herald Lab has a long-standing expertise in retrogenic mouse models, and by using CRISPR technology, we can generate kind of custom-made novel knockout combinations in effectors and initiators of cell death, as well as set up in vivo screening platforms for host genes critical for restricting intracellular pathogens. But some of you might wonder, oh, this is all very interesting, but why is it so important to dissect those cell death fail-safe pathways during infection? Because, of course, knowing these fail-safe systems can identify an Achilles heel for the pathogen and inform how to therapeutically target cell death to eliminate intracellular infections. And this is, of course, where Mark is the puppet master, and work in our lab has previously shown that this can be done successfully to promote clearance of HPV, for example, but also malaria. And the lab has also generated some very exciting data and soon to be published data on tuberculosis, as well as HIV and HTLV using humanized mice. So watch this space. And as shown just now, knowing that the host depends on different cell death modalities to kill parasite infected cells as well, we're also interested and already have begun to exploit this in Leishmania, Toxoplasma, and Cryptosporidium disease model. And furthermore, as I've been talking a lot about salmonella, we think that targeting cell death in salmonella infection could also be an interesting avenue to tackle the problem of chronic salmonella carriers. And in some really preliminary work I've conducted, treatment of salmonella infected cells with BH3 mimetics kills the cells by apoptosis and thereby degrades all cellular contents, including the bacteria, which is reflected by a significant drop in bacterial colonies that remain viable when plating the remains of the cell. And Sami Bedoui's PhD student Sven, who was a great help during the project already, will now look into the possibility of using BH3 mimetics to induce killing of salmonella infected cells in vivo using the mouse model that I have previously described. All right, now last but not least, I want to point out that the models we have established can be utilized to answer open questions on the role of caspases beyond infection control and even beyond cell death. And it's my wish to fulfill Andreas' big dream, to generate cells without any caspases. And this would enable us to look into the survival function of caspases, or their role in differentiation, proliferation, and to clarify long-standing issues about the role of caspases, for example, in metabolism, or as a mediator of post-translational modifications. But we're also extending the portfolio of knockout mice, and I'm currently breeding mice that are lacking all initiator caspases. And those mice, we can then interrogate the role of caspases in development and cancer. All right, so with this, I'd finish my talk, and of course, thank first and foremost Marco, Andreas, and Mark, as well as Sami Bedoui from the Doerflinger Institute, who have been central in driving this project. Of course, Yeshuan and Rania, who have been with me from the get-go. Paul Whitney and Sven Engel from Sami's group, who did a lot of the in vivo work. Many people here at WeHi that contributed with experiments or reagents and mice. The girls in bioservice, because they were taking care of all the mice. Collaborators internationally as well as nationally, the WeHi community, friends and family, and some of the funding bodies that contributed to this work. But I'd really like to end with who are the real masters of puppets are. And I know that Marco is a great Metallica fan, and this might be one of his favorite albums ever. So Marco and Andreas were the puppet masters together with Mark and Sami and driving this project. And with this, I think I have a few minutes left for questions, and I'm happy to take them.
Thanks very much, Marcel. Yep, you do have quite a few questions. I'll start with, does Salmonella secrete effectors that could mediate cell death independently from host proteins?
Yes, I think I scratched this point a little bit towards the end when I kind of wrapped up why it's so important to know how all these cell death pathways are being regulated. Because indeed, Salmonella has effector molecules, and so do a lot of other pathogens. They have these effector molecules at their disposal, and that's the point I wanted to make, is that really we evolved so many functionally overlapping cell death pathways to counteract those proteins that the bugs have on their side.
Thanks. So Hamza Puthalakath asks, like, why did Typhoid Mary's macrophages not die?
That's a good question. So we know that there is chronic human carriers, of course, and so maybe, you know, in this case, the bugs found ways to persist over and above bugs that infected other people. So maybe they have effector molecules that have given them the opportunity to persist. And hopefully we can address this by tackling the Achilles heel of the intrinsic apoptotic pathway.
Good. Can you expand on the method of BMDM immortalization and comment on any effects this process might have on cell death mechanisms? Did you confirm the results in primary BMDMs on human cell lines? So I think you did address that in the...
I did address that throughout the course, yes. I mean, these immortalized bone marrow macrophages, of course, as that would definitely be in a review question. We had to make sure that these cells can actually die, and I showed throughout the talk that their death response really correlated to what we saw in the primary bone marrow cells. We haven't utilized human cells to confirm this, and it would be a lot of work to use the same CRISPR combinations to do so. But we definitely think that in human cells we would see a similar pathway overlap occurring.
Another question to have is what happens to caspase knockout, RIPK3 knockout, etc. cells loaded with bacteria since they can't kill themselves?
What would happen to them in vivo? Is that the question?
Well, I suppose even in vitro, I presume they would just continue to accumulate the bacteria, but something must happen.
I mean,
would it be necrotic death that they undergo?
I think eventually they will undergo some kind of necrotic death if we would have left them for longer than 24 hours, because I guess at some point they're really swamped with bacteria to the absolute maximum. And I showed that there was no induction of ER stress after 24 hours, but we haven't really looked into those cells much longer than that. And we know that when we infect these cells with really, really high MOIs of bacteria, that eventually we do see some cell death happening. So it's some kind of overt cellular stress that eventually will drive the cell into killing themselves by some way. But this is definitely not physiologically relevant if we infect with super high MOIs in the Petri dish.
I think we've probably only got time for one more question, and it's
make it a good one.
Well, it's actually a question that Anne Voss has provided, and one that I have always asked you many times, and that is, what happens to the salmonella within the cell undergoing pyroptosis? Are they always effectively phagocytosed, or can they also be released given that pyroptosis involves cell lysis? And have these different possible fates been assessed or quantified?
Yes, and I think that's a point I also made towards the end, is that we always thought that the inflammatory caspases per se induce pyroptotic cell death, but we think, given that caspase 1, that our work showed that caspase 1 can do many other things, including inducing apoptosis, that it might be a trade-off in vivo, where some cells are being purged in order to recruit more immune cells and to activate certain arms of the immune system, whereas mainly the cell, you would assume, would drive itself into apoptotic cell death, because with this the bacteria are contained and degraded. So I hope I could answer this question good enough, but I think it's really difficult to really get to the bottom of this in vivo.
Well, thanks very much, Marcel. There was about 4 or 5 other questions, some of them really quite intriguing, from Seth and Charlene and Andre. So if you wouldn't mind having a look at those, I've published them.
Absolutely, yes, sure.
Thanks very much for the presentation. It was really quite amazing to see all the data and the meticulous lengths you went to knock out absolutely everything you could. Thanks again, and thanks for joining us, everyone.
Thank you.